just some disclosures. I've served as a scientific advisor, a consultant to Psychedelics and a group called Brainify, but I won't be presenting any data related to my work with these organizations today. I will present a little bit of data related to an academic collaboration with David Olson's lab, but that's kind of separate. Jumping in, as Cam just mentioned, I'm a neuroscientist and a psychiatrist, and one of the big questions that we're interested in my lab is the mechanisms that mediate mood state transitions over time. So depression, this is kind of stating the obvious, but I think sometimes we overlook this fact, which is that depression is by definition a fundamentally episodic psychiatric syndrome. It's defined by periods of low mood interposed between periods of wellness. And there are interesting individual differences in the temporal dynamics of people's mood episodes over time. And again, anyone who's a clinician in this room knows this and sees this every day. But there are very few studies, longitudinal studies, tracking patients over time or tracking animals as they kind of cycle in and out of these depression-like behavioral states. This is one exception to that. This was work done by Bob Post and his colleagues like 25 years ago now, where they tracked individual patients with unipolar major depressive disorder and bipolar disorder and just described the dynamics of their mood states over time. And you can see three examples here. Periods below the x-axis denote periods of depressed mood. Periods above the x-axis are hypomania or mania. And zero represents stable, normal euthymic mood. And I think you can just see by just glancing at these three individuals that they look very different. I think that's the main take-home message here. The first individual, for example, I have an arrow here. Can you see that? That's okay. The first individual has kind of characteristic durations of their depressive episodes, at least at first. Whereas the second individual has much shorter episodes, but also shorter periods where they're well. And Post and colleagues identified still other examples that looked quite different as well. And we really don't understand much, frankly, about the mechanisms that determine, for example, why a person becomes depressed today as opposed to last week or next month. And when a person gets depressed, what determines how long they stay depressed? And conversely, when a person gets better, what are the mechanisms that initiate that recovery? And what are the mechanisms that sustain it over time? Why is it that some individuals get depressed just once in their life, get better, and never get depressed again, whereas other individuals on the opposite end of the spectrum struggle with depression on and off every year for most of their lives? And, you know, undoubtedly many mechanisms are involved. We've been interested in pursuing this in a variety of different ways, but the general approach in my lab has been either to do repeated longitudinal neuroimaging fMRI studies in human subjects, with or without treatments, and try to understand what changes in the brain, what might kind of precede and predict the onset of depressive symptoms, and what changes when people get better. And likewise, we do repeated imaging assessments at the microcircuit level in mice. And this being a conference focused on psychedelics, I'm gonna tell you mostly about our work in that second area, looking at preclinical models of depression-related behavior and how they're altered by ketamine, a rapid-acting antidepressant. And I'll finish up by telling you a little bit about similar mechanisms that we think are involved in psychedelic analogs. As I said, we think probably many mechanisms are involved, but converging data from a variety of sources suggests that one of those might involve the remodeling of these structures called postsynaptic dendritic spines, which you've heard about earlier today multiple times, I think first from David and then from others. Spines are these microscopic protrusions, which you can see pictured here from the dendrites of neurons. You can think of spines as being kind of structural markers of a synapse. Most spines contain synapses. I'll be using the terms kind of interchangeably today, although it is the case that not all spines contain synapses, and we could talk about how those are different if people are interested. We know that in various chronic stress models, which produce behaviors in rodents that are reminiscent of what we see in some depressed patients, we know that there's a loss of dendritic spines in the prefrontal cortex. And conversely, we know that antidepressants like ketamine tend to do the opposite, increasing the density of dendritic spines in the prefrontal cortex. So it's been tempting to speculate that these two things are causally related, that changes in spines might lead to changes in behavior. But it's also possible that they're just correlated with some third factor. And really getting at cause and effect is important, not only for academic kind of scientifically interesting reasons, but also for real like practical translational ones. If we think that the formation of new synapses is causally driving changes in behavior, then that has all sorts of implications that are different from a model in which they're just correlated with something else that's actually driving behavioral change. And so in the first kind of set of experiments I'm going to describe, we set out to ask these 3 questions. This is work led by Rachel Moda-Seva, an amazing grad student who's now at Harvard, Mitch Murdoch, who has now moved on to MIT, and Pooja Parekh, who's a postdoc in the lab and will be starting her own lab soon. They set out as a team to ask these 3 questions. First, does ketamine, a rapidly acting antidepressant, selectively rescue stress-related synapse loss? I think you've already seen data showing growth of synapses. I'm going to tell you a little bit more about what precisely these new synapses might be doing, and how they might relate to changes that we see in chronic stress states. Second, we'll be asking how the formation of these new synapses influences circuit function and behavior. And then third, we'll look into that crucial question of causality. Does the formation of new synapses drive the behavioral change? And not to kind of bury the lead, I'll just tell you that we found that they were causally important, but not in the way we expected. And I'm going to explain that in a little more detail over the next few minutes. So just to familiarize you with the kinds of methods that we're using before I jump into the data, we're going to be imaging the remodeling of postsynaptic dendritic spines in the prefrontal cortex of mice in the living brain. And in order to do that, we need to gain optical access to these deeper brain tissues in mice. The medial prefrontal cortex, does this pointer work? Yes. Lies buried deep in the medial wall outside the range of conventional 2-photon microscopy, which is about maybe like 500 micrometers below the surface. And so to get to these deeper brain areas, we implant a microprism into the brain. We can fix it to the skull. The mice tolerate this procedure very well. It has some unique advantages. Although we're doing some damage to the hemisphere in which the prism is implanted, the rest of the brain is structurally intact, and we're imaging along an optical path reflected into the opposite hemisphere where the cells are intact. The second advantage is we can get a lot of light in and out of the tissue. And that means by returning to using like vascular landmarks in the contours of the prism, we can return again and again to the exact same positions, and that's what you see photographed in the lower right. And we can ask kind of with micrometer precision, which spines are disappearing after chronic stress, and where are new spines forming after ketamine treatment, and how do they relate to one another. And so in this first set of experiments, Rachel and Mitch implanted a prism. They gave the mice some time to recover, and then they used 2-photon imaging before and after various chronic stress models and asked what impact that had on spine remodeling. And what they found, and incidentally, what we'll be looking at here is depicted in the images at the left. The blue and red arrows denote eliminated spines present in the first image, but not in the second. And the white arrowheads denote formed spines present in the second image, but not in the first. And so what they found first was not altogether surprising. Chronic stress increased the elimination of dendritic spines. But because we're doing this in vivo, we can begin to ask more targeted questions about whether there's a logic to the spines that are being lost during chronic stress exposure. We did that in 2 ways. One of them is shown here. So we reasoned that if you assume that particular synaptic projections to the prefrontal cortex are being targeted for pruning and others are being preserved, then you might expect that lost spines would tend to cluster together on specific dendritic branches. And that's because we know from others' work that one of the functions of dendritic branching in the prefrontal cortex is to segregate functionally homogeneous synaptic inputs. So all of most of the inputs from region A might tend to synapse on the red branch 1, and most of the inputs from region B might tend to synapse on the orange branch 2. And that's indeed what we found. You can see on the left, the distribution of lost spines across dendritic branch segments was highly bimodal, where about half of branch segments exhibited spine loss rates comparable to controls, whereas the other half exhibited really markedly elevated spine loss rates, sometimes up to 10-fold the normal number. Not shown, we also found that lost spines were highly spatially clustered. They tend to co-occur on branch segments all bunched up right next to each other. What about ketamine? So here, the exact same mice were imaged before and after chronic stress. Then we treat them with ketamine or a vehicle control, and we ask what impact that had on spine turnover. Essentially no effect on spine elimination, that's not shown. Instead, what we find is something very similar to what I just described, but instead of for eliminating spines, ketamine is forming new spines. It's doing so in a bimodally distributed, branch-specific way. We also find that they're spatially clustered, kind of popping up in the same little bits of dendrite. And so that led us to ask whether it might be the case that ketamine is actually acting in some targeted way to restore spines that were lost during chronic stress exposure, restore them in the same position. Or is it the case that they're just popping up in random places? And so to test that, we classified each spine that formed after ketamine treatment as a restored spine if, like the ones in blue, it formed in a position where a spine used to exist, but then disappeared after chronic CORT. Chronic CORT is a neuroendocrine model of the response to stress. We see similar effects in other stress models. Or a de novo formed spine, if like the one in purple, it formed in a position where we didn't observe a spine in either of the previous two images. And the upshot is that we found that ketamine had a big impact on spine restoration. Many of the spines that were lost after chronic stress, they disappeared and they reappeared in the same position after ketamine treatment within like 2 micrometers, which is really pretty remarkable. That effect was much larger than you'd expect by chance based on the observed rate of spine formation. We think it's a real effect. And this is interesting. And it's kind of like, before I move to the functional effects, I kind of look at this as like the glass half full version of what I just described. But I also want to emphasize the glass half empty view, which is that although many spines are being restored, it's not like all of the spines that form are restored spines. Some of them are popping up in random places. And what that means is that many of the spines that were lost after chronic stress are popping up again, but some are not. On average, about half of the spines that disappeared after chronic stress are restored and half are not. And then some of those won't survive indefinitely. So it really is kind of a partial rescue is the way I like to look at it at the level of spines. And we think that might have implications for the reemergence of depression-related behaviors later. And I'll return to that idea in a couple minutes. So moving to the second part, I told you we would be interested in understanding what effects chronic stress and ketamine were having on circuit function and behavior and how they might be related to the changes in spines. And so in this series of experiments, we started by doing something very similar to what I just described, but instead of imaging spines, we're imaging circuit function. We do this by using 2-photon imaging in mice that have been genetically engineered to express a genetically encoded calcium sensor, GCAMP6S. The upshot is that this probe fluoresces when calcium pours into the cytoplasm, meaning that when the cell becomes more active, it glows. And it gives you this kind of Van Gogh starry night appearance of cells talking to one another. And we have algorithms that others have developed for taking these images, extracting single cells from them, and you can get calcium transient time series from each cell, which is what you see depicted on the upper right, 4 examples of that. One of the predictions you might make if many synapses are disappearing after chronic stress is that the structure of correlated activity, what you can think of as functional connectivity between these neurons, might change, and that ketamine might reverse that effect. And so that's the thing we tested first. And to do that, we did kind of 2 different analyses, which you see in a representative mouse here. In the lower panels are correlation matrices where warm colors denote pairs of cells that are strongly functionally coupled, strongly correlated. Cool colors denote weaker or no coupling. And what you can see in the baseline condition compared to the chronic CORT condition in this very same mouse, that there's a lot less warm colors. You can just kind of don't need fancy statistics. There's a lot less functional coupling. And another way of viewing that is in the top panels. These are raster plots. Left to right is time. Left to right is time. Each row is a cell, and each element is colored based on how active the cell is at that time. And what you can kind of see, again, just by glancing at this, is that a lot of the activity that occurs at baseline occurs in the context of these multi-cell ensemble events, where many cells become active at about the same moment, sometimes even most of the cells in the field of view. Interestingly, these events seem to be rhythmic, which is something we didn't necessarily expect or design the experiment to detect. I can return to talking about that if people are interested. But what we do know is that chronic court reduces the occurrence of these events. And when they do occur, they tend to involve fewer cells. And again, I don't think you need any fancy statistics you can just kind of see it looking at the data. We, of course, did quantify it statistically. It's present in all of the animals we looked at. It's very robust. And we find that ketamine imaging the same animal one day later rescues both of those effects. Okay, so that's interesting. What does all that mean behaviorally? So there are kind of circuit functional correlates of the changes in spines that I described, but are they behaviorally relevant? So we looked at this in a number of different ways, and this eventually turned into a bunch of different papers, and I don't have time to describe all of them in detail, but I'll just highlight like a few examples. So we've been looking at the role of these ensemble events in driving motivated escape behavior in response to a threat, in cognitive flexibility and kind of adaptive reward seeking, which I'll describe a bit. Also in social hierarchy perception and motivated reward seeking behavior. I'm just going to show you, this is 5 separate papers, 25 figures, a lot of data. I'm just going to show you a few highlights, and I'm happy to talk about any of the details at the end if people are interested. So the first thing we looked at was motivated escape behavior in the tail suspension test. This is motivated by really beautiful work from Melissa Warden, who showed that ensembles of prefrontal cortex neurons that project to dorsal raphe, the nucleus in the midbrain that controls serotonin signaling, when you optogenetically drive them, you can change behavior in tests like the tail suspension test. The tail suspension test basically just asks what a mouse or rat does when confronted with an inescapable threat. And you'll hear this interpreted in different ways. And the way I like to interpret it is just kind of in a conservative way that's anchored in what we're actually observing. The mice alternate between periods of struggling to escape and freezing. And the kind of just so story that people have for why they do that is that mice are kind of fundamentally prey animals. Everything that a mouse meets is a potential predator, pretty much. Even rats eat mice. And so if you're a mouse, many of your predators rely on vision to catch you, and a potentially adaptive response to a threat is remaining perfectly immobile so that maybe you don't get seen. And another good response is trying to flee to get out of there as quickly as possible. And that's what mice do in tests like the tail suspension test and the forced swim test. And we know that chronic stress increases their tendency to be immobile. And we know that antidepressants tend to do the opposite, which is also driven by stimulating these cells. And so we wanted to ask what ketamine was doing to this behavior and to this, this circuit in the context of these kinds of assays. And essentially what we found is depicted on the upper right. This is a photometry recording from ventromedial prefrontal cells. This is a mixed population of ventromedial prefrontal cells. And you can see during periods of struggling in pink compared to periods of immobility in white, the activity in this circuit tends to be higher. But that might be related in a kind of non-specific, uninteresting way to just motor activity. And so we honed in on another interesting observation, which was that the increase in activity tended to precede and predict shifts from immobility to struggling very reliably. And that's what you can see in those images at the left. The increase happens first, and then about a second later, the mouse starts struggling. And we see that over and over again. And essentially what we find is that chronic stress reduces the occurrence of those events. And so there are fewer shifts from immobility to struggling, and ketamine rescues that effect. Again, suggesting in a correlative way that the ensemble events that I just described might be behaviorally relevant in the context of depression. We've also been looking at other arguably more interpretable behavioral assays that are also sort of more complicated. I'll highlight just a couple of examples. We're very interested in how these circuits encode and support effortful reward-seeking behaviors. All of us, when we encounter a reward in our environments, have to kind of make a decision whether to work to pursue that reward or not. And that's going to be influenced by our expectations about how much the reward is worth and about how much effort is going to be required to obtain that reward. And what we find is that, so this is one example of a test where we can measure that. Mice are trained to navigate in a T-maze, turning left or right to get a large reward or a small reward. But to get the large reward, they have to climb over a barrier. And what we find is that cells in this circuit, these are prefrontal cells that project to the nucleus accumbens, they encode an interesting signal, which anticipates the acquisition of a reward, but also scales with the amount of effort required to obtain that reward. Bigger rewards mean a bigger signal. So these neurons are integrating information both about reward magnitude and effort requirements. And if you optogenetically silence these cells after the mouse has made its decision, you actually influence its future decision making. It stops making high effort, high reward choices. And interestingly, we don't see that at all if you silence it before it makes the decision. So it seems like it's a kind of post hoc reinforcement signal for future decisions to use effort to obtain a reward. We find that chronic stress diminishes that behavior and also disrupts that kind of effort sensitive reward predictive signal in these cells. And we're currently looking at how ketamine influences it. I'll show you one more example, and then I'll return to the kind of key question of what role spines are playing in all of this. We heard multiple times today, including just a few minutes ago, about the role of cognitive flexibility in depression and how it might be modulated by psychedelics. We've been studying this using a model in mice. The behavior we use is, so this is a paradigm developed by an amazing postdoc, Tim Spellman, who's now running his own lab at the University of Connecticut. Tim developed an automated kind of high throughput behavioral task for mice that is meant to mimic the Wisconsin card sorting task, which you heard about earlier. This is kind of complicated. You probably got the gist of that before too. But the upshot is we're recording from hundreds of prefrontal cortical cells through a prism while mice perform a task in which they receive combinations of whisker stimuli and odor stimuli. And they learn over a series of trials that the odor is what they're supposed to pay attention to. The odor predicts how they're supposed to respond, lick left or lick right, to get a reward. Until eventually the rules change on them. And I'm oversimplifying a lot here, but eventually the rules change and in what we call an extra dimensional attentional set shift. Now the mouse needs to stop attending to odor and start attending to the whisker stimulus. And it will learn that, but it takes some doing, just like it would in a person. It takes mice a little longer, but they learn it. And this task, we know that it requires prefrontal cortex function in both humans and rodents, but we don't know exactly what kinds of information these cells are encoding and what they're doing to support the behavior, which I think is kind of a critical foundation for understanding how antidepressants like ketamine and psychedelics influence the function of those circuits and how we might tweak them. And so summarizing a ton of work that Tim did to pursue this, he found kind of first confirming that the prefrontal cortex is required for this task. If you inactivate it with a drug called mucimol, the mice are much slower to learn the set shift that I just described, the rule change. In the middle, you see, and this is essentially what we predicted, but it was kind of interesting, the strength of this signal. In the middle, you see that these neurons in the prefrontal cortex are encoding information about all sorts of task features, basically all the task features. You can decode from the neuronal activity what the mouse is observing, like which odor has been presented, which whisker stimulus has been presented, what the current rule is, whether or not the mouse got rewarded, all sorts of information about the task. But interestingly, and this is not what we expected, when you optogenetically silence these cells during task performance, there's actually no effect on the behavior. That's very puzzling because there's a lot of literature suggesting that this structure is required for the task. And in our own hands, I showed you that if we inactivate it with mucimol, you disrupt their performance. So this is credit to Tim. He was able to kind of rethink our model and figure out what was going on here. And after a lot of work summarizing like a whole bunch of figures, what he found out was this. So it turns out that these cells are encoding a kind of continuously updated working memory-like representation of the mouse's recent behavior, what it chose, and whether or not it was rewarded. And that's what you see on the left. So this is our ability to decode trial history on a given trial based on the activity of these cells. And if you inhibit these cells, not during the trial itself, but after the trial is over, that disrupts performance. And interestingly, it disrupts performance only on trials that are what we call incongruent. They're trials that are informative as to what the current task rule is. So as you heard in one of the earlier talks and in what I just said, this task is uncued. You're just getting rewards or not rewards. And so no one tells you that the rule has changed. You have to figure it out by yourself. And the mice learn that some trials are informative as to rule and some aren't. And inhibiting this signal after those trials disrupts performance. We also find that this behavior, not shown, this behavior is stress sensitive. And again, we're looking at how ketamine and in collaboration with David Olson, how some psychedelic analogs influence function in this test. And I'll return to a little bit of that at the end. Yeah, I'm going to skip this and just get to the kind of key question, which is whether synapse formation is really required for inducing these antidepressant behavioral effects after ketamine treatments, or just correlated with them. And again, this is not what we expected and turned out to be pretty interesting. So we asked this question in two ways. One is kind of like brute force, but also kind of simple and elegant in its logic. Like if you think that A causes B, then looking at the time series of events will tell you something about causality, because if A causes B, A has to come before B. And if instead you observe that A comes after B, that tells you something right there about cause and effect. And so no one had ever done that. We should have anticipated this result if we looked a little harder at the literature. No one had done that exactly, but there were other hints. And what we found was that in an experiment where we imaged mice before and after chronic stress, then repeatedly for hours to days to up to a week after the ketamine treatment, we found that the effect of ketamine on behavior was rapid. You could detect it just 3 hours after treatment. We now know, I think David showed you some data on that too. You can see this 30 minutes to 1 hour after treatment, a change in immobility behavior on the telesuspension test. The effect on spines, in contrast, is slower. It doesn't emerge until 12 hours after treatment. It peaks a day after treatment, and it persists to some degree for a week. And so what that was telling us is that the formation of these new spines actually couldn't be required for initiating ketamine's antidepressant effects, at least not in the cells that we're imaging from. It must be something else. Interestingly, when we looked at the time course of the effects on circuit function, we found that they paralleled behavior and preceded the effects on spines. And that's what you see here. The emergence of the effects on correlated activity in these prefrontal neurons in the lower panels is detectable 3 hours after treatment. Same for the reemergence of these ensemble events well before we can detect any change in spines. So does that mean that the spines have nothing to do with the treatment? This is kind of puzzling at first. And so we went back again to kind of rethink how we were viewing this. And one hint came from this scatter plot from the same experiment, which showed that the restoration of lost spines, you recall, we operationalized restored spines as spines that form after ketamine treatment in a position where a spine used to exist and then disappeared. We find that the restoration of lost spines was really tightly coupled with the maintenance of ketamine's antidepressant behavioral effect assessed 2 days or 1 week after treatment. And so it suggested to us that maybe it's the case that ketamine's forming new spines, and that isn't required for initiating the behavioral effects, but perhaps it's required for sustaining them over time. And so to get at that question, we teamed up with a group at the University of Tokyo led by Haruo Kasai and Haruhiko Bito. They developed an optogenetic tool, which they call activated synapse photoactivatable RAC1. The name is kind of a mouthful, but the tool is very powerful and simple and elegant in a lot of ways. This is a blue light sensitive probe whose expression is targeted to a sparse subset of newly formed synapses and newly potentiated spines. And excuse me, upon prolonged exposure to blue light, it changes the conformation of the probe and initiates the RAC1 cytoskeleton, initiates the RAC1 signaling cascade, which through interactions with the actin cytoskeleton causes spines that express the probe to shrink and to collapse. So the upshot of this is that it's an optogenetic tool for selectively deleting in a very selective way newly formed spines and synapses. And Harua and Haruhiko and their team used this probe to test whether the formation of new spines in the forelimb area of motor cortex was required for the learning of a motor skill or the memory of a learned motor skill. And what they found was that it wasn't required for the initial learning, but it was required for maintaining a memory of the motor skill over time. And we wanted to ask whether something similar might be happening with ketamine. And so we did an experiment where we exposed mice to chronic corticosterone in their drinking water, a model of chronic stress. Spines disappeared. We treated them with ketamine. Some of those spines came back. We photoactivated this probe 12 hours after ketamine treatment. And then we asked what impact that had on behavior 2 days or 1 week later. This is just validation data showing you that this intervention was sufficient to disrupt, kind of reverse the effects of ketamine on spine formation. And then the critical behavioral data is plotted here. It shows you that chronic cort in red increases mobility in this task. Ketamine reverses that effect in blue. But if you delete those new spines in green, the maintenance of ketamine's antidepressant behavioral effect, it wears off. They look almost like the stressed animals that didn't get ketamine. We also had an important negative control where we photoactivated 3 hours after treatment in light green. You'll remember that 3 hours after treatment was a time before any new spines formed due to ketamine. So what we're doing here is deleting another kind of random set of spines that are not related to ketamine treatment. And what we find is no effect on behavior. So I think what this tells us is that the formation of new spines, new synapses, we do have data suggesting an equivalence of those things in this context. The formation of new synapses after ketamine treatment is required not for initiating ketamine's antidepressant effects, but it is required for sustaining them over time. And those new synapses are doing something special. They may be related to sustaining the effects on those multicellular ensemble events and some of the coding functions that I described that prefrontal cortex is so important for. And I'll close by describing briefly a collaboration. This is unpublished data with David Olson's lab, asking whether spine formation after treatment with a psychedelic analog tabernanthalog is serving a similar role. And so this is data from David's paper. Yeah, it's not showing. Yeah, it is. Okay. This is data from David's paper led by Lindsay Cameron, published in Nature in 2021. They imaged cortical neurons before and after treatment with tabernanthalog. You'll recall TBG is an analog of ibogaine, a psychedelic compound that has these amazing behavioral effects, including changes in depression-related behaviors in David's study. And we wanted to know whether the formation of these new spines, like what role were they playing in the context of TBG treatment. This is just really nice validation data from David's paper showing that, that TBG is increasing spine formation in these neurons and having correlated effects on tail suspension test behavior, immobility behavior, that seem to be serotonin 2A receptor related, because if you co-treat with ketanserin, you don't observe those effects on behavior. And so teaming up with David's group, this is work led by a postdoc in my lab, Shane Johnson. We just did an experiment very similar to what I described for ketamine, treating mice with TBG, waiting 12 hours, photoactivating with the probe to delete the new synapses that form after TBG treatment, and then asking what impact that had on behavior. And what you can see very clearly, even in a relatively small cohort of mice, is that photoactivation effectively reverses the effects of TBG on tail suspension test behavior. So I think what this suggests is that both TBG and ketamine and perhaps other antidepressants may be acting through sometimes similar mechanisms, sometimes different mechanisms that may converge on the formation of new synapses in prefrontal cortex, and that the formation of new synapses in turn is required for maintaining those effects over time. And that's really important because we know, and we heard this from multiple talks today, including the one you just heard, that the effects of ketamine, I don't mean to undermine it, they've been life-saving for many people, but it's not a panacea, right? It's not like it cures depression. It's a really powerful treatment for some individuals, but we know that absent some other intervention, you treat people with ketamine, they're not going to stay better forever. You need to do something else to help them stay better. And these data suggest that other interventions aimed at boosting the restoration of lost synapses or prolonging their survival over time might be useful for augmenting the durability of drugs like ketamine and TBG. And we're currently looking into that. And I think with that, there's plenty of time for questions still. I'm just going to stop. I just want to highlight my lab members, especially the people highlighted in blue, who did all this work, and my collaborators, Hiroka Kasai, Haruhiko Bito, and David Olson, who also made this work possible. And I think I'll stop there.
Thank you for sharing those wonderful data, and I'm really excited to see some of the crossovers and corollaries with cognitive flexibility. Really exciting. I got a couple of different times you presented data that suggested that if you provide some kind of a block or kind of antagonism or something like that, after a learning trial, that it would kind of interrupt the learning. And I guess, is there any relationship to the idea of reconsolidation or blocking reconsolidation or blocking some kind of memory or learning process that we have? Is there anything to flesh out there?
Yeah, that's a great question. We've been interested in that too. Like rephrasing, essentially, we find that the formation of new spines isn't required initially for learning, but is required for retaining a memory of that learned experience. And perhaps those mechanisms might relate to really well-studied mechanisms at both the molecular and circuit level that support the reconsolidation of memories. Reconsolidation, and I'm not an expert in this area, but for those who don't know the term, refers to a process when a memory is retrieved for kind of consolidating the trace of that memory in a way that helps to kind of solidify its durability over time. And yes, we actually think that they probably are related. So we've been looking, for example, this is not published yet, I guess. We've been looking, for example, at fear conditioning and extinction learning, and the reconsolidation of extinction learning after retrieval, and using this same optogenetic tool to delete spines. And essentially, we find, just as I think you might predict, that the formation of new spines is important for maintaining a memory of the learned event over time. If you delete them, it doesn't interfere with the acquisition of conditioning, but it does interfere with retrieval or with storage over time. And if you retrieve, it looks like the spines become kind of labile and prone to being able to be deleted again within some window, and deleting them will interfere with that. Great idea.
Hello, and thank you for the wonderful presentation. I don't mean to oversimplify something that is pretty complex, but I've heard talk today about loss of dendritic spines, restoration of dendritic spines. Can you comment on any research on too many dendritic spines, any pathological potential or therapeutic potential of that? Thank you.
Yeah, absolutely. So the question, I like that. We've been focused today on the idea that forming new spines is a good thing, and probably it is a good thing in many contexts. But are there contexts in which you can have too many spines and synapses? And I would say probably yes, there are. So there are neurodevelopmental conditions that are characterized by changes in the mechanisms that kind of regulate the development of spines early in life. And as summarizing a lot of work, kind of the normal process is that early in life, you have this big burst of spine formation. You have lots and lots of spines, probably too many, more than you need. And then there's a normal kind of physiological pruning process that occurs. And people think maybe that pruning is helping to kind of sculpt your brain to the particular circumstances of the environment that you live in. And so both of those things, the formation and the pruning, are normal kind of healthy things. And you can imagine how if either one of them went awry, it might lead to problems. And I am not an expert in this area, but my understanding is that there are neurodevelopmental conditions that are associated with both too much or too little, or either too much or too little spine pruning or spine formation. And the upshot of that can be actually at certain developmental periods, an abnormally large number of spines that seems to correlate with learning problems. So I think like the bottom line, your question's really well taken, is that like although we're talking a lot about spine formation, it is a balance. And having too much or too little of any of these processes is probably not a good thing.
Hi, Connor. Thank you for that very elegant work and nice talk. I have a couple questions that are very much related. We think of spines as being the structural correlate of a synapse. And the fact that you're able to see the synapses return in the same spot suggests that maybe the synapse was not completely eliminated, and that all of the structure needed for the synapse still is retained. So my question is, one, is there evidence that these synapses are truly lost versus maybe becoming less relevant? And two, along those lines, do you see if you do, I don't remember, it was 12 days of cortisone injections. If you prolonged that, do you get less return of these synapses in those same spots? Is there a temporal correlation between how long the stressor is there and the ability of those spines to return?
Yeah, those are both great questions. So with respect to the first question, spines are popping up in the same place. How do they know where to pop up? That's sort of like what you're asking. And one idea is that some remnant of the synapse might still be there, but not visible, or perhaps a fully functional synapse is there, but we just can't see it in the form of a spine. There are limitations to the resolution of our microscopy. All points well taken. To address that, we did a lot of stuff. We do believe that probably some of those caveats are at play to some degree, but we also believe that synapses really are being lost. So like one way to look at that is postsynaptic density staining. So we find a big reduction in PSD95 after chronic stress and a restoration, the opposite effect after ketamine. So it does seem like grossly there's effects on synapses, not just spines. Doesn't rule out what you said though. We also know that new spines, spines that persist for more than 2 or 3 days, almost all of them contain functional synapses. So one way of getting a sense of whether effects on spines also pertain to effects on synapses is through longitudinal imaging. And we know that through those experiments that ketamine is forming new spines that persist for days and do presumably contain functional synapses. How do they know where to come back? I think your idea that there may be some remnant on the postsynaptic side is probably one of the mechanisms. Could also be the case that there's some presynaptic partner. We're only imaging the postsynaptic side here. The presynaptic side is invisible to us. It may also be the case that neighboring spines emit diffusible factors that kind of regulate and control where new spines can form. We know from a number of people's work that that's the case, and those mechanisms might be involved here too. We're trying to figure out like which, if any of those, is involved and what the synapses are connected to. We don't know answers yet. Your second question was about, it's getting a little late for me and my working memory is not working. Yes, the length of the stress. Yeah. I love that experiment. We haven't done it. So if you prolong the stress, like the hypothesis would be if the stress was long enough, you'd get to a state where ketamine, its ability to restore spines is kind of attenuated or abolished. And I would guess that the answer is like, maybe to some degree. So we know that like with repeated exposure, this is an experiment we have done, like a lot of the spine loss occurs relatively quickly. So there's sort of like, a lot of the spine loss and then like diminishing marginal additional spine loss with more days of cord exposure. So I would guess it's not like twice the amount of cord exposure is twice the effect. It's probably not at all the case. But it stands to reason that like, yes, the more spines you lose, the harder it may be to restore the circuit. And we really should look into that. I like that idea.
Yes, thank you. I wanted to just reference Dr. Spellman's work. Muscimol was being used. And that was interesting to me because that's a psychoactive ingredient in Amanita muscaria mushrooms. And it looked like, I wasn't sure if that was being used as a comparison to ketamine. We're going by a little bit quickly. So what led to the choice of muscimol? Why that's psychoactive?
Yeah, that's a great point that I glossed over. So muscimol is a GABA agonist that's commonly used in mouse studies like this. We infuse it directly into the brain, and it transiently inactivates neurons in the area to which it's infused. So it's like historically has been kind of the tool that people use if you don't want to lesion a brain area, but you want to like turn it off temporarily. But great point. Muscimol is also like a psychoactive compound when taken orally, probably at higher doses. That was not the purpose that we had in mind when we did this study, but it's an interesting thing to think about. Yeah.
Conor,
Yeah.
I have a question. I'm maybe making too many connections, maybe because that's like thinking. So you showed for the PFC that there was a disruption with chronic stress. There was a disruption in this computation, which could be like an expected value computation, or something like that would be involved in the motivated reward-based decision-making piece.
Yep.
And is that's thought then to be a substrate for depression, sort of local circuit manifest or prefrontal manifestation of depression-related behavior?
Yes, that's our hypothesis. Yep.
And so then you've got the spine finding with ketamine and psychedelics, and the notion that there has been learning and that it's involved in the consolidation or reconsolidation of that learning. So what's driving the learning? What's the missing link there? That's the thing that's being reconsolidated, that's fixing the disruption of the expected value computation in the prefrontal cortex.
Yeah. That's a great question too. And we've been working on that for 4 or 5 years. We think we have something approximating an answer now, sort of like what initiates the changes at the circuit level that are being consolidated by the formation of the new synapses. So I'll give you our working model right now. So it's kind of interesting. We started this thinking, this is going to take me a minute or two to describe. We started this with a couple of hints. We know from Ron Duman and others' work that interneurons seem to be involved. If you inactivate, knock down NMDA receptors in different interneuron classes, you'll interfere with the effects of ketamine on behavior. And we also know from work from folks at Stanford that if, which might have come up earlier today, that if you co-treat patients with naltrexone and ketamine, that you block the antidepressant effects of ketamine, but not the dissociative effects, interestingly. And so that got us interested in whether a mu-opioid receptor mechanism, perhaps involving interneurons, might be involved. And basically what we find, summarizing a lot of data, is that I hesitate to say that it's the mechanism. One important mechanism is probably GIO signaling in somatostatin interneurons. The mu-opioid receptor is selectively kind of overexpressed in somatostatin interneurons compared to other neurons in the prefrontal cortex. The mu-opioid receptor is coupled to a GPCR, which initiates GI signaling in SST cells, and mu activation essentially silences the SST cells, disinhibiting pyramidal neurons and restoring those ensemble events. And we think that's a very rapid thing, like seconds after infusion. And we think that's probably one of the key initiators of the antidepressant effect at the circuit level, that in turn later leads to changes in spines, which are required for sustaining it, and enables neurons to re-engage in this ensemble activity that's so important for example, coding these like effort and reward sensitive signals. Thanks.